All right, excellent. So I'll echo back and say I appreciate the opportunity to speak here. And I'm actually calling in from a workshop exploring this at the USDA, the National Wildlife Research Center in Fort Collins, Colorado. And there's folks here from Island Conservation. This is an NGO that I'll talk a little bit about, and CSIRO in Australia, and USDA personnel, both research scientists and regulatory personnel, and trying to investigate this. And I also say that this will be much less technical because I think things have not, at least in terms of these applications, progressed nearly as far along as they have for insects. So a lot of this will be, why would you consider this, for example, for islands? And so talking about the current methods, the limitations, but also the potential benefits if you succeed with this. And so I'm from NC State University in Biological Sciences and also part of the Genetic Engineering Society program that Fred maybe mentioned a little bit at NC State, and he and Jennifer Kuzma are, of course, directors of. So just in terms of organization, I'm going to provide a quick overview of the problems presented by rodents in a pest situation, falling in three major areas: so economic costs, acting as disease vectors, and then what we've been focused on, biodiversity threats on islands. And we look at this both as an important problem in its own right, as well as these islands potentially providing, containment in some respects, and so in that sense, potentially being useful test site opportunities. And then talk a little bit more about what we're currently doing for eradications on islands, the benefits of that, but then also the limitations. And then again, just really relatively briefly at the end about some of the things we're pondering for genetic control strategies for rodents. Okay, so starting off with some of the economic and food security impacts, just as an example, the estimates range of pre-harvest losses for rice production in Asia range from 5 to 25%. In some cases, these can be borderline catastrophic, 50 to 75%. And in rough terms, and these are figures from the International Rice Research Institute in Grant Singleton, this is enough rice to feed 180 million people. And this is probably conservative because that's all pre-harvest and not post-harvest losses being included. And those can be, you can see it indicated there, a good 25 to 30% in India, it looks like, and probably somewhere in other places. Of course, these rodents can also be important disease vectors for a whole variety of things. And, obviously very famous from medieval Europe, but continuing today in other respects. Okay, focusing on islands more specifically, and the sort of why would you consider doing this question. Islands are pretty special in terms of biodiversity, so they're only about 5% of the land area on Earth, but they contain about 2% of bird, reptile, and plant species. And if you just look at IUCN critically endangered, critically threatened and endangered species, we're talking about 40% of species. And so the history of this has been pretty bleak on islands. In terms of known extinctions, we're looking at anywhere from 70 to over 95% of the known vertebrate extinctions have occurred on islands. And so we've been fortunate to work with this non-governmental organization, Island Conservation, and they're a very focused group, and their exclusive focus is removal of vertebrate pests from islands where they present a threat to biodiversity. And a conservative estimate that they've put together is that there's a major conservation threat on at least 1,300 islands worldwide. And there's a real data gap here. It's probably much larger because this is a figure that always strikes me. It's estimated there are about 180,000 islands in the world, and if 80% of them have rodents, then we may be looking at a threat on up to 150,000 islands. So for this reason, there have been a lot of eradication efforts, and we've made an awful lot of progress. And this is focused on a variety of species, mostly animals, but in some cases plants as well. And this is a graphic where the animal's body size is roughly proportional to the scale of the threat they present. And the major threat, you'll notice actually those are not mice here, which is mostly what we're focused on, but rather rats. And we've been thinking about mice because, as it turns out, mice are harder to eradicate than rats for various reasons I'd be happy to talk about. And mice are also more tractable for studying. And so what we learn in mice could potentially be applied to rats. But most of these efforts have been focused on rats. And just to give a couple of examples of what these look like, here is, I think, the largest one to date. And it was the eradication baiting phase completed this past March. But this is the island of South Georgia, about 400 square miles, or 100,000 hectares. It was really an extraordinary operation, so multiple years. You know, these are the estimates on just what it took in terms of supplies: 750 helicopter loads, 200,000 kilos of bait. And this was facilitated by the fact that this is a subantarctic island. And so it's actually divided up by glaciers, and it gives you some manageable chunks. But still a pretty heroic undertaking. They're now into the monitoring phase, which tends to go a couple of years. You know, you're looking for any evidence that you've missed a rodent. So these are challenging, of course, because essentially missing one pregnant female is a 100% failure for the operation. So these eradication efforts have to be very complete. So what are the benefits of that? Why would you do it? So it's been actually very encouraging. Effects at every level of the ecosystem on these islands. The impacts can be really dramatic. And oftentimes, I mean, we're starting to get an understanding of what happens just based on the fact there have been over 350 eradications to date. But we can see things like forests reappearing, and they can be quite different than what you saw there before. Many of these invasive rodents especially are very hard on seabirds. There are stories from Alaska finding rats on piles of over 100 dead seabird chicks. And in the Galapagos, where there have been some recent operations, and I'll skip to the next slide here, species are reappearing that were thought to be lost. In some cases, species that were never known to be there have been discovered. And then species that are quite iconic are doing a lot better. And so these are shots from Isla Pinzon in the Galapagos, and Island Conservation, in collaboration with other folks, ran this operation down there. And so this was in late 2012, and by about a year later, the first hatchlings, these iconic Galapagos tortoises, were seen surviving in the wild for the first time in perhaps 150 years. So the results can be dramatic and very positive if you prioritize biodiversity, as Island Conservation does, and it's a big part of what really drives us in this. So, but these are really challenging operations, and they don't always go up. And so here's one a little bit closer to home, the Farallon National Wildlife Refuge. And so this is, if you look out through the Golden Gate in San Francisco on a clear day, which is rare, you can see them off in the distance here. And it's a very rugged place, remote, the kind of place that is really good for seabirds, though. And there's very little in the way of human habitation, just a couple of structures, and they house people from Point Blue Conservation Science, which used to be the Point Reyes Bird Observatory. So the threat there is posed by mice, and the focal point, if you will, of that is the ashy storm petrel, which is about 60% of the world's breeding population breeds on Southeast Farallon Island. And the nature of the threat comes in a kind of a complex way from the mice. There are some direct threats, but primarily it comes from the effect they have on the behavior of this animal, the burrowing owl. And so this figure just shows, it illustrates this, what's termed a hyperpredation phenomenon. So in red, we can see mouse trapping success, and so this is a strongly seasonal population. They get to terrific numbers, perhaps the most dense population known from natural habitat for house mice that has been recorded in the world, 500 or 600 per acre at their peak. And so that's tracking the mice, and then around about November, December, they start to crash because the weather turns bad, meet some food restrictions, and actually these predators, the burrowing owls shown in blue, arrive. And where this really becomes a problem from a conservation standpoint, in addition to other things the mice are doing that are less obvious, is there's a, to use a bad pun, the burrowing owls encounter not a movable feast perhaps, but a moving feast. And this encourages them to hang around, if you will, for longer. And they stay long enough that they're there when the ashy storm petrels arrive to breed. And they start depredating ashy storm petrels. And the projections are that this could severely endanger this bird over the longer term. So that's the rationale. that the Fish and Wildlife Service has for doing this. And they have an environmental impact statement for proposed eradication using rodenticides. It runs for over 700 pages, very carefully thought out. Many years of effort has gone into it, but they're running up against a real challenge with off-target effects, which I'll get to in just a second. In this case, the likelihood of high mortality of gulls, also potentially other non-target species on the island, and at least at this point, it has delayed this operation. So just to get into some of the challenges of this method, so these rodenticides can work quite well, but it's a major and challenging operation. For anything over about 5 hectares, you need certainly toxicants, but also helicopters, and it runs into a lot of money, which I'll get to. The tool that's being used is a second-generation anticoagulant. There's something called verdificum is the one of choice. It's highly toxic to rodents. It's been developed for that purpose, but it can also be quite toxic to other things. There's one operation, for example, in Alaska that was very effective against the rats, but it resulted in the death of a number of bald eagles, and so that's obviously not desirable. These are very challenging operations, and this is really from talking to one of the participants here and working with him, Carl Campbell of Island Conservation. Even on uninhabited islands, this is very, very difficult. And the complexity, the cost, regulatory challenges, everything goes up if you add people to the mix with livestock and pets and children. And to the point where we're just starting to get into that, but it's the sort of thing that is expensive, it's difficult, much harder to arrange even than these other different operations which that you have to do in the absence of people. Okay, so some continued limitations. What we're doing now is it becomes very difficult because you have to essentially apply these baits with a little bit of spacing over the course of about a week or 10 days. And they have to be applied very precisely in high doses because, again, you can't miss a pregnant female. And if the weather turns on you or the season is, if you have, say, an El Nino year, it's not favorable for this, you're perhaps spending $60,000 per day by the time you're set up and ready to go and you've contracted helicopters and things. And so it adds up fast. And so if you look at the bottom here, they're working on an operation for Floriana Island in the Galapagos. It's estimated at this point to come in around $14 million. Macquarie Island, south of Australia, about $26 million. And it takes a long time. It's about 9 years from inception to completion. And so part of the reason I bring all this up is, you could say, why would we contemplate something like the genetic engineering approach? And it's because we do have this other approach, but it has a lot of limitations and downsides. And just continuing on some of those barriers, again, one of the major ones is it's not species specific, and so this is where genetic approaches could offer real benefits. I mentioned the inhabited islands, real challenges there, acceptability of these methods. An animal dying from a rodenticide is it's pretty gruesome, really. So they die of organ failure and blood, a lack of ability to have blood clots and things, and so internal bleeding, and it's just not a pretty way to go. So there's concerns, reasonably, about applying toxins in the natural environment from the public, and lethal methods generally are just not. They have real downsides in terms of public perception. Okay, so what are some of the things that are being pondered from a genetic engineering standpoint? And again, this is, I think, very, very early days, and perhaps early days even for some of the insect work. Those folks could comment on that, but much more so for this. The overall idea would be to prevent female development, and in that sense, reduce the ability of the populations to grow. And these animals get into an almost exponential growth curve, and it leads to these extraordinary population densities over short periods. And so if we can prevent development of females, or conceivably prevent sexual maturation of those females, or the ability to breed, we could interfere with that cycle. So 2 possible ways to do that. One would be inducing male development through the introduction of transgenes. And so a benefit that we have with mice is they're perhaps the best studied animal we have, and we know an awful lot about the process of sexual development in mice, with a great number of genes that are necessary for this and can disrupt it. And one of the most important ones, and the one that David Threadgill's lab with Donna Canavy as a graduate student at Texas A&M is working with is SRY, which stands for sex-determining region of the Y chromosome. I'll show you a little schematic on the next slide. That's the master testes-determining gene in mammals and across mammals with some exceptions, and you get outside the eutherian mammals. So that's one possible approach, and we also know that about a variety of genes that are critical for female development. One that we, for example, pondered that is important as far as we know across vertebrate animals is something called FOXL2. It's a 4-kit box transcription factor in an adult female mouse. If you essentially knock out its function in the adult female, her ovary will start to become a testis in many respects. And so there are these possibilities. The technologies that you would use to do this, David Threadgill and Donna Canavy are exploring right now, is a naturally occurring meiotic drive system in mice called the telele. And so if we can put in a SRY transgene into the telele and have that inherited in super-Mendelian fashion, and result in no fertile females, that potentially could be a good option. And then, of course, since even we started thinking about this, CRISPR-Cas9 has come on the scene and creating all sorts of other options, but all sorts of other challenges. Okay, so this is just a quick schematic of what this might look like with this SRY mouse. So they carry SRY on the Y chromosome, but you'd add this into an autosome. You have a mating with a wild-type female. This would result in 2 kinds of offspring, theoretically. An SRY male, so it would have really 2 copies of SRY and will be fertile, and an SRY XX individual that should be a sterile male, at least based on previous research. Okay, so lots of challenges, of course. First thing is you could have the world's best genetic construct, but if those males can't successfully compete and mate in a natural environment, this is not going very far. And that's one of the things that in my laboratory we're really focused on, trying to ask that next step question, if you will. Could these mice be competitive? This will require testing under realistic but also very secure conditions. And really want to emphasize this workshop we've been having, a very careful staging of this approach. And one of the reasons we're here at the National Wildlife Research Center is they have really superb facilities that might enable testing under realistic natural conditions, but in a very secure way, so high level of biosecurity. Of course, the other challenges arise in the area of how do you regulate this, and how would you go about even testing this from a regulatory standpoint. And so it's been wonderful because we have the folks here who are real experts in this for pesticides, and right at the very beginning, and they've provided a lot of really excellent guidance that way. And then I think the challenge for all these things, of course, is how do you effectively communicate and engage the public? And as we don't want to all of a sudden, of course, appear, say we've got this great mouse, and it's going to cure everything, and have people feel perhaps surprised by that. Okay. Keegan had also asked me to just say a little bit about the timeline. I would say that's very challenging to predict in this case for a number of reasons. The technology on this has changed an awful lot, as everybody here is aware, over the last couple of years, and the likelihood of continued changes is, I would say, pretty high. We're talking an absolute minimum of 5 years testing that would be necessary to have an idea whether this could work, moving up in scale, and I mean, certainly no plans perhaps even on the horizon, but there are places, for example, where you've got remote oceanic islands that have invasive mouse populations, and they're relatively small, they don't have connections to other places, and one important thing I think from a containment standpoint is we do have methods in place that are effective for eradication. And so you could terminate that experiment. And so that the methods for doing so are pretty well understood, I think. From a regulatory standpoint, 5 to 10 years might even be optimistic. I mean, there's a lot that needs to be figured out here, but that also seems to be the case for a lot of aspects of this. So this is obviously not in isolation, so it'll be interesting to see how that develops. And then, again, back to the communication and engagement. Do we start now and continue through testing and implementation? I think that would be a good idea where possible. So just I'll close by acknowledging some of the, again, the workshop partners here, Island Conservation, the National Wildlife Research Center, which is a division of the USDA, Texas A&M University, where David Threadgill, his lab are based now, CSIRO from Australia, and NC State University, where I'm from. And if people are interested, there's a few other slides on why this might or might not work, and an awful lot of questions. So thanks for your attention, and thanks again for the invitation.
Thank you so very much, John. So again, we have a few minutes of questions for if the committee would like to ask John Godwin some questions about his presentation. Remember to unmute yourselves and go ahead and ask us questions. Also, our speakers have done a phenomenal job with keeping ahead of time. So if you have questions for either Zach Edelman or Fred Gold that you were not able to ask earlier, please do ask them questions after we spend some time with John.
Hi, this is Lisa Tannehill. John, I really enjoyed your presentation, and I was wondering if you could elaborate a little more on what types of containment measures or regulation and safeguards you think might be necessary when you're doing this research both in the lab and later in maybe controlled field trials.
Mm-hmm. Yeah, so that's a challenge, and I would say that we have a lot more. I'll just start off by saying I think we have a lot more thinking to do on that, and that's part of what we're doing today at this workshop. All those folks are on the other side of the wall here talking about it. So that's one of the reasons I mentioned that we came out to the Wildlife Research Center. They have really a pretty spectacular facility, and they do a lot of animal disease research, for example. They have worked with transgenic rodents, and one of the really appealing features here is they have rooms that are I think very nicely sealed up, which is important because mice can get out through small spaces, and that are about 1,000 square feet, and they can generate natural illumination, climate cycles. They can even make it rain in these rooms. So hopefully we can do tests under something approaching natural conditions as an intermediate step from smaller enclosures still, in order to ask, could these mice be competitive? Look at a sufficient number of mice to ask, how might a drive system work? Would it actually accomplish what we think it needs to accomplish? And there's going to be a lot of things that need to be addressed there. Again, we know a fair amount about what makes mice competitive breeders, but there's a lot we don't know. There's some anecdotal things that suggest this could be promising, and social dominance is a big one, for example. And actually, genetic dissimilarity turns out to be an advantage for a male mouse, and so introducing a mouse that could benefit from that. So back to the containment at essentially a middle step here might be a place like this, and we hope it's a place like this, where they have experience on this, where they've thought an awful lot about how to keep animals in, but where they have this at such a scale that you can start to ask this in a hopefully realistic way. We know, and there's many examples of where these animals will do well in shoebox containers, for example, or you won't actually see a phenotype, but then you expand it to something approaching semi-natural, and you have dramatic effects. And so we can't test this the way we do many things with rodents. And then the next step on that containment, again, I'd mentioned there's Carl Campbell has identified some candidate islands in Australia, which may be the logical place to do this for different reasons. They have a variety of islands that could be good test cases. They have a regulatory structure that may be easier to navigate. In order to develop this, it's a little simpler than our system. And on those islands, I mean, one aspect of containment is they're remote. In many cases, there's not a dock, there's no people there. In some cases, you can have access that's tightly controlled. And again, because we're at about 360 eradications now worldwide on islands, and we do have the tools with these anticoagulant rodenticides, and we know how to apply them to terminate that experiment. And so I think that's an important aspect of it. There's some other things that have been suggested by Kevin Esfelt, for example, where you might first drive a unique sequence into a population and then base your gene drive on that. And so in theory, at least, you wouldn't have, although Zach's talk gave me pause on this, in theory, at least, you would not have possibility of that invading, say, a mainland population. So I don't know if that answers your question, but those are the sorts of things we've been talking about.
Great, thank you.
Any other questions for John? Or Zach or Fred? In that case, thank you, Fred, John, and Zach. Oh, is somebody?
Hi, this is Yuri Holzer. I have a quick question or maybe a comment for Fred. Going back to the non-sexually reproducing populations, I wonder if we are maybe unnecessarily pessimistic about our ability to use some of these tools, because let's say going back to the coffee berry borer, we know that even these what we look at as non-sexual populations, they do have the capacity to fixate alleles under strong selection pressures. For example, the coffee berry borer has a history of fixing insecticide resistance across vast areas if that insecticide is applied routinely. And so there is probably some minor amount of outcrossing, but it is significant if the environmental pressure is also significant. And so I wonder if rather than driving in a single feature, if we use some of those combinations of features being driven in, if that's possible to use even in populations that are mostly selfing, but then we would give them an advantage if they're not selfing. Does that make sense?
I think Fred is muted. Hold on, just give us a moment. Fred, we've unmuted you if you'd like to respond.
Can you hear me?
Yeah.
Okay. Yeah, I think that's a good point. I think that it would be useful to investigate. But I think you're talking about two somewhat different things. One is with the insecticide resistance, you're talking about a very strong advantage to the mutation and that inbred population. So you would expect to see a signature of selection, where you may have a selective sweep for those populations moving into, taking over. Whereas when you're trying to decrease the population, you're selecting against those individuals that get that. So you have it's not the analogy. The analogy is not quite the same. But I think it would be useful to investigate it. But I think when you look at the population structure of the coffee berry borer, it does look quite strong. So it would argue that this would be difficult. But I think you bring up a good question. It would be worth looking into. I certainly wouldn't want to do too much with that when you have so many better targets to look at to start with.
Any other questions from committee members for our speakers?
Yes, this is Liz Heitman. I have a question related to the charge for the committee, which is to talk ultimately about what responsible research looks like in this field. And just the three of you speaking today demonstrates how complex a field, if it is in fact even one field we're talking about. But if we could ask each of you, if I could ask each of you to say something about what you think the most important, and I put this word in quotes, ethical charge for people working in the area in which you're working, and perhaps what you think your most important ethical commitment is in the work that you're doing. That could be very helpful in directing how we interpret the large quantity of information that you were able to distill into today's presentation. And that was not meant to stop conversation.
I guess I can go first since I spoke first. That's a really important and difficult question because there's very different levels at which we're talking. There's the ethical issues in terms of looking at the costs and benefits, and when you think about something like malaria. And I brought up the tsetse fly issue where you're balancing things, where you have to deal with justice. I mean, there's a huge number of things that need to be looked at even before you start deciding that you're going to do a project. So I think that's important. And I think sort of this issue of, making decisions in a democratic system. You need input. I think a lot of you would say, you can't just decide on these things as the researcher, because you have a bias to start with because you want to have a successful project or something like that. But then there, I think, are other issues in terms of just moving along once you decide you're going to work on something. And I think Kevin Esvelt and others, Akbari's paper, are important to consider in terms of how do you make sure that you're not going to have an inadvertent release. I'd say that the Gates Foundation, in an earlier work on Aedes aegypti in Mexico, had a good example of how they interfaced with the country and their regulatory officials to do a step-by-step process. And I think you could find good literature from those works. Janine Ramsey has published on that. And the work that they're doing now with the malaria in terms of taking a very methodical approach to make sure that you have buy-in by the people who you're working with and that you've examined the potential downsides. So that's what I could think of off the bat, but I know it's a very complex system.
Yeah.
I think that's the big challenge of why they called us together, is how complex it is.
Yeah. Good. You do that work for us.
Thank you. Yes, we're on that.
So I guess I can chime in my 2 cents, and I'm going to echo everything that Fred said, so I don't repeat myself there. But really, just the concept of the regulatory structure that's built up in this country is really not up to the challenge, and I think that's been recognized. But I just want to echo that, is that our whole regulatory structure is based upon containment. And how do you keep containment? And Gene Drive is the antithesis of that, because we're releasing it and we acknowledge that we don't have containment of it. So I think that just some kind of new structure needs to be developed, where all these different complex things can be fit in to a certain pathway, but that pathway has to be predefined. So it's not everybody in the Wild West doing things on their own, but that we have a structure that people can follow. And some things will be more difficult than others from that regulatory structure, but it needs to be developed so that we have a clear path of, here's a safe way to do this, here's a path forward, and for the various different types of Gene Drive, that path might be more arduous or less depending on how controllable your Gene Drive system is, and I would like to see just more in turn kind of a feedback type of research about how these can be further improved, how they can be further controlled. Can you eventually have something that's is capable of very strong Gene Drive, but that is ultimately very controllable also? And I don't think that we're there yet. Even with the hypothetical things that have been put forward, as I mentioned, they don't remove Cas9, they don't remove a nuclease from the population. They never really do get back to wild type. And so how do you revert in the case that something has happened? I think that will go a long way towards being able to satisfy a lot of regulatory and social issues as far as we can pull this back, because it's a very scary thought to say we're going to do this and it's unstoppable. I think that's a barrier that we need to have both strong regulations for, and then further research to figure out how to create systems that are strong but are stoppable.
I really like what you just said about the idea that Gene Drive is the opposite of containment, because we have been talking about that system.
So I guess I'll chime in here at the end. And I'd like to echo what Zach just mentioned about our regulatory system as it is, perhaps not being up to this. And it's been really an interesting conversation here at this workshop with these folks who are deeply immersed in this with the USDA. And, you know, if you have a modified mouse, what exactly is that? And it looks like under, you know, current regulations, it's an investigational new animal drug in technical terms. And who regulates that? Probably the Food and Drug Administration. You can do consultations with the EPA, but it certainly has the feel of a jury-rigged system from that standpoint. You know, and we have to, it would be certainly good to rethink this, given these organisms that can be produced now. I'd say the other part of this that we're hoping will develop this group here, and it's great to have the participation. We have Peter Brown from CSIRO, is the involvement of the Australians, and it certainly would be good to see other countries involved as well. So we're obviously not in isolation here. And so I think it's going to be an arduous process even within a country, but across countries. We can start moving towards some sort of consensus on this, and how do we properly regulate these things? How do we properly contain them? And I would say the one other thing, just going back to your original question here, what is a responsible approach does this look like? I think probably the watchword is cautious, carefully staged, with attention to issues like containment at every step. And along with that, you know, as transparent a process as we can have. So, I mean, public perception is of course enormous on this, and so hopefully we can safeguard that public perception. And I thought to myself, I mean, I wish in many ways that people could be on some of these meetings that I've been in this past week and what you folks have been doing, and just to see that, you know, I think we are genuinely concerned about this. And, you know, there is real potential here, but if we have a misstep, we could derail things that could be quite promising and could do an awful lot of good. So, I mean, it's incumbent on us, both for the good of the eventual project, but also just, you know, like Fred said, we live in a democracy, and it's important to have that public engagement, public approval. So I'll leave it there.
Thank you.
I'll bring up just one more thing, and that is just to say that the first few that are done have to be done incredibly carefully, because if there's any mishap, as John said, some mishap in the beginning, it could get in the way of doing real good for society.
Thank you.
Hi, this is Lisa Tannehill again. I wanted to ask one more science question to all of you. One of the things that Kevin Esfelt has been promoting is this idea of a split gene drive that would be used with Cas9 and the guide RNA being sort of on different cassettes. And I was wondering if you could discuss whether you think this type of split gene drive system would be feasible in the various model organisms that we've been talking about.
Well, I can go first. As far as feasible, that remains to be determined, and I'll speak from a mosquito point of view. But as I showed, a split gene drive still has a lot of the issues that you find in a normal gene drive. That is, the guide RNA or the Cas9 protein and guide RNA can still recognize non-target sites. And without defining what those sites are or creating your guide RNA in such a way that there are no off-target sites, you don't really know what's going to happen, and you can end up driving other things in the population. You know, I think that was basically for laboratory experiments, if I remember correctly, because then you could have something escape, but then that would not spread itself in the wild because the Cas9 could not replicate. That is, in a laboratory colony, you could have a fixed Cas9 expressing strain, but the guide RNAs would drive themselves through the laboratory colony, and in an escape, the wild population would not have Cas9, so you would block the escape. So that's a kind of a method for containment, but I'm not sure you could use that to drive something in the wild, although, again, Cas9, because it does work in trans, does not need that to be there. So as a biocontainment, you can do it, And a lot of those things are designed for laboratory biocontainment, but I'm not sure about in the wild whether that's what you want to do. But you could have, you know, as long as you take into the same things into account, what is happening outside of my target site, then that's just something you need to investigate on a case-by-case basis. I'm not sure if that answered your question or not.
No, that's fine. Thank you.
Any other
Can I
questions?
just pop in? I would, you mentioned, I actually, this is John Godwin, I don't feel qualified myself to answer that question on the mouse side. They're obviously very engineerable, but that would, I think you'd want to probably talk to some other folks about that. But it's an important question, certainly. I'll leave it to Fred now.
Well, I think Zach made the important comment that this is good for laboratory containment. I think it could very well work. We are building population genetic models to look into ways that we could use the CRISPR-Cas9 system with the expectation of a certain period of time for disintegration of drive. And having them not linked at all might not work, but there might be some in-between approaches.
Any last questions from the committee? Okay, with that, I'd like to thank our presenters and for everyone listening on the phone joining us today. You get almost 20 minutes back to your day, and stay tuned for our next webinar, which will take place on next week on October 21st. We will be hearing from Ethan Beer and Valentino Gantz on their research on gene drives in Drosophila. So thank you again.
Thank you all very much.
Thank you.
Thank you.